My lab has been working in two general areas to delay aging. And the first we started when Tori Hagen, a brilliant postdoc, came to my lab back in 1995. And I had been interested in DNA damage causing cancer and how to delay it. I got more and more interested in preventive medicine. And then I realized that cancer is one of the degenerative diseases of aging. That even if we stopped smoking and ate better diets, we'd still have cancer because it goes up with the fourth or fifth power of age. And then I became very interested in aging, and more and more I became convinced that mitochondria were something to get your hands on. Sitting around waiting for a rat to age for 2 years or for 3 years or a mouse to age in 2 years was just too long at my age. And so we decided that mitochondrial decay, there was a fair amount of evidence saying that oxygen radicals from mitochondria were damaging the mitochondria and causing a good part of aging. So we decided to work on that, and Hagen knew how to do it. So our strategy, so one of the side products of, as mitochondria get old, they let out more and more oxygen radicals. As you know, mitochondria burning your fuel, which means pulling electrons from them, and adding 4 electrons to oxygen, and then you're home safe to water, and that process puts a potential across the mitochondrial membrane and allows the cell to make ATP. So there are 500 or so mitochondria in every cell, and you make kilos of ATP in your body, and so all the fuel you burn is burnt in the mitochondria, and all the oxygen you breathe in goes through the mitochondria. And there was a fair amount of circumstantial evidence, and we wrote a big review on the subject, pointing to mitochondrial decay driving a lot of aging. Parkinson's is associated with complex 1 decay, and Alzheimer's with complex 4 decay. So we set up all the assays in mitochondria from old rats that declined with age. And then we did interventions in the rat to see if we could find substances that could delay that. And we found 2 mitochondrial metabolites, acetyl, carnitine, and lipoic acid, that together could solve all these 4 decays, and then we published a lot of that. And I'll only very briefly go over that because I'm more enthusiastic about what we're doing now, and all this was published 6 years ago and before then. So this is mitochondria, the outer membrane, the inner membrane, and you have each of these complexes which have hundreds of genes. 1,000 genes are made in the nucleus. There's a little barcode. The protein is sent into the mitochondria, and the mitochondrial DNA makes a few proteins. So now what you're doing is you're burning your fuel, and in that process you pump protons across the inner mitochondrial membrane, and that charge across the membrane allows complex 5 to make ATP. And so mitochondria are basically rechargeable batteries. It's the charge across there that allows you to make ATP. And with age, what happens? Well, cardiolipin in the inner membrane, it's only used in mitochondria, a key lipid, goes down with age. It's one of the most easily oxidized fats. And then oxygen utilization goes down with age. The membrane potential goes down with age. And then the byproducts of all this, oxygen radicals, go up with age. So we found that acetylcarnitine solved 3 of the 4 problems. It didn't solve the problem of the oxygen radical goes up. And we got onto that because some Italians had published a paper, Garoletta and Paradis, that they fed acetylcarnitine to old rats, and mitochondrial transcription got better, and we had all these terrific assays, and we tried it, and it made 3 of the 4 decayed functions better. And then we looked around, we tried lots of things to see what would reverse the increase of oxygen radicals with age, and we found something that worked, and that was lipoic acid. We found several things that worked, but one would have had to be a drug, and we were looking for nutraceuticals, so we found lipoic acid worked, and that solved the problem of the oxygen radicals. And as I made the mistake of telling some reporter after our paper came out, those old rats got up and did the Macarena. So that appeared in the news stories all around the world. And then I got a very angry letter from some Scottish pharmacologist who chewed me out for about 6 pages that it was very unscholarly. The rats didn't really get up and do it. And I should be ashamed of myself. And I wrote him back a sheepish letter. It was a joke. But anyway, so I won't go into all the biochemistry. We showed all kinds of things. But I'll just show you. We did a lot of tests in old rats on IQ and all of that, and doing IQ tests in old rats is a pain in the butt, but we did it. And this shows the Morris water maze, where you have a little pond and turbid water and a hidden platform, and rats can swim perfectly well, but they don't like to swim with nothing underneath them. And you have visual cues all around. The first day you put the rat in the pond and you show them where the hidden platform is. Next day you throw them in and keep on measuring. Well, young rats make a beeline for the hidden platform. This is the time to reach the hidden platform, and old rats wander all around the place. And acetyl carnitine helped a little bit, the lipoic helped a little bit, but the two together were better. And then people said, well, it's just their eyes going out, it's not the brain. So we did another test, the Skinner box, and a psychology professor at Berkeley had done all sorts of neat tests where the rats have to measure a time interval. You put them in a little box, and if they press a pedal at the right time, they get a food pellet. So we triggered it by sound, and we triggered it by light. It was identical. It was a synergistic interaction between the acetyl carnitine and lipoic and making it better. And then we did it in mice. And then the next thing is we did it in dogs. This shows, there's a company in Canada called CanCog. I don't know whether it means canine cognition or Canadian cognition, but they do tests on old beagle dogs and young beagle dogs. So, and the university had taken out a patent on this combination, and I had formed a company with a businessman friend of mine called Juvenon, and they sell these pills over the web. I put all my stock in a nonprofit foundation and given away all the money to science. So anyway, but so I get no financial interest in this, so I can mention it. Anyway, Juvenon paid this CanCog to do some IQ testing in dogs, and we got an excited phone call, best thing he'd ever tried, and the dogs made many fewer errors if you give them acetyl carnitine and lipoic. So that's all I want to say, except that we published showing that nucleic acids in the brain get oxidized, and the combination helps. This shows we had a monoclonal antibody that reacted with both oxidized RNA and DNA, and this shows the hippocampus, young rats, old rats, were staining DNA in the neurons in the brain, and this is the combination. But we got a bit of a shock because the nucleus wasn't staining very well. Well, we knew DNA got oxidized, but most of the oxidation, as you heard from Christian, is in RNA. And so we added RNase and DNase. We struggled a little bit, John Conley did these experiments. We struggled a little to find an RNase that didn't have DNase, but we finally found it. And RNase takes away the stain, DNase doesn't, but there is some damage to DNA too. And then we worked on mechanism. Tori Hagen showed that when he was in my lab, that glutathione went down with age, and lipoic acid brought it right back. And so we thought lipoic acid was just becoming the reduced form in mitochondria, and that was acting as a terrific antioxidant. But it didn't turn out that way. It turns out that lipoic acid is a fantastic inducer of all the body's antioxidant defenses, the phase 2 enzymes, 250 enzymes that protect sulfide group from oxidation. And Jung Soo in Hagen's lab published some beautiful papers on it. So it's better to turn on your body's defenses than to just add an antioxidant. Well, that's all I want to say about that. With the money from the companies going into clinical trials, we've had one successful clinical trial on hypertension, and then more and more coming along, and I'm very optimistic. Now I'd like to talk about why you're all aging yourself faster. So And that's because you're eating bad diets. This is in general. I'll give the reason why. So the case I'd like to make is that there are 40 micronutrients. These are the substances you need to power your biochemistry besides your fuel. There are about 15 minerals, magnesium, manganese, iron, copper, you know them all. And about 15 vitamins, most of which are coenzymes, but some of which are hormones. And then 2 essential fatty acids, the omega-3s and the omega-6s, and then about 8 essential amino acids. And so we've been showing that deficiency of all kinds of micronutrients gave you DNA damage and makes human cells and culture zinc deficient. You get oxygen radicals pouring out of the mitochondria. You get massive DNA damage. You knock out P53, so you're getting another whammy, and you knock out copper, zinc, SOD. So it's a very efficient way of giving you massive DNA damage. And we showed it with iron. We showed it with biotin. So we must have run it through a half a dozen or more micronutrients, and they all gave DNA damage when you made it human cells and culture deficient or mice deficient. I said, what the hell is going on? Why is nature doing this? And then it hit me, that's exactly what nature wants. Through all of evolution, animals have been running out of one micronutrient or another. The 15 minerals aren't spread evenly through the world. Some areas are high in iron, some are low. Some are high in selenium, some are low. So what happens when an animal is running out of a magnesium, for example? It's going to die. So nature selects for short-term metabolism at the expense of long-term metabolism. So you survive and maybe can reproduce. So anything long-term, the metabolism gets cut out ruthlessly when you're short of micronutrients. You may look perfectly healthy, but if you don't get your magnesium, we're talking about more than half the country, you're knocking out your DNA repair enzymes, and you're knocking out your adaptive immunity, and your mitochondria pouring out oxygen rapidly, all the things that normally come with age. That's the case, and more we're finding evidence for that, and I'd like to just briefly discuss that. So anyway, this is metabolism. So we've cured beriberi and pellagra and scurvy and all those diseases, and people forgot about vitamins. But there are really a lot of people who are very low. The nutritionists have the committees making the EAR and the RDA. The RDA you all know about. It's the recommended dietary allowance, and that's the amount the population should be getting. Well, there's a measure of population inadequacy called the EAR, and that's the RDA is 2 standard deviations above the EAR. So if you're below the EAR, the committees say, well, the population below the EAR is inadequate. But nobody gives it that, because here we have 56% of the US population is below the EAR for magnesium. Nobody cares. All the poor, all the obese, a lot of the elderly, teenagers, 80% of them. Where do you get magnesium? From your spinach. It's in the center of the chlorophyll molecule, just the way iron is in the center of the hemoglobin. You eat a big plate of spinach, you're getting a big dose of magnesium. Americans aren't eating big plates of spinach. And you can also get something from whole grain bread. So, but if the pathology is really there and insidious, which is what I'm going to argue, then we need to look in a different place. And I think the place we have to look is DNA damage, and we're in the middle of clinical trials trying to look at that. And it's adaptive immunity going out and mitochondria pouring out oxygen. So I'll go through why I think that. Now, 16% of American menstruating women don't have enough iron. 2 billion women and children in the world are too low in iron. We heard from Christian that iron excess is bad for you, but iron deficiency is a complete disaster in the world. And so the World Health Organization goes in and they give them iron. They're giving them too much iron, but I'll discuss that later. So, Anyway, and then men are getting too much iron because they're eating too much red meat as adults. Zinc deficiency, 12%, and Emily Ho in my lab showed zinc deficiency. It knocks out P53. P53 is a zinc enzyme that's mutated in half the human tumors. Well, you can do it by mutation. You can also do it by not getting enough zinc into you. So you're disabling all your DNA repair systems. And then zinc deficiency also causes the mitochondria to pour out oxygen. And then B6, half of elderly women. Folate, half the pool was low in folate, and so they added it to the flour because of neural tube defects, but we still have 16% of adult women low in folate. E, practically everybody. C, a third of people. And then omega-3 fatty acids, the whole country is short of those. And African Americans with a dark skin are very short of vitamin D. And you can go through this list. Sometimes I think Walter Willett's the only guy in America who's eating the perfect diet. He's the chair of nutrition at Harvard who does all this lean everything option. But anyway, that's a slight exaggeration. So we're trying to figure out now whether we're talking about 20% of the US population or 80% of the US population. I suspect it's more than half. Anyway, we'll hopefully know soon. So I got onto all this because Jim McGregor came into my lab, and he's a cytogeneticist, and he was measuring chromosome breaks by something called a micronutrients test. And he was irradiating rats and get more broken chromosomes. And then one day, his technician came in, and all his control rats were full of broken chromosomes. And he said, "What's going on? Did you irradiate them by mistake?" It turned out someone had left folic acid out of the chow of these mice. And folic acid, so they did a lot of dose response and everything, and they showed that, in fact, folic acid deficiency is just like radiation. It breaks chromosomes. And so, I put my graduate student and postdoc to work it out because he had just come to my lab on sabbatical. And we showed that this is the key pool of folate. It methylates uracil to make thymine. And thymine's in DNA and uracil's in RNA. So this pool goes up, this pool comes down, and you start putting massive amounts of uracil in your DNA. Now the base pairing is the same, but the repair enzymes are cruising along the DNA looking for trouble, and they're constantly taking out uracil, so you get nicks in your DNA. Now the radiation biologists think radiation works because it gives you clusters of electrons, and the most dangerous part of radiation are double-strand breaks, chromosome breaks. Because you get two nearby lesions, because you get a cluster of electrons and two oxidized bases. And normally when you have a lesion in DNA, the repair enzymes come along, they see something that isn't right, they clip it out, they put the right base in, so and the other strand holds it together, so they sew it up. But when you have two nearby lesions, your repair enzymes are actually doing you in, because they take out this one and this one about the same time, and the chromosome falls apart. Anyway, that's the going theory of the most dangerous part of radiation. And folate deficiency does the exact same thing. Now, McGregor looked in people, and the method you use in rats and mice doesn't work in people, but he managed to find some people he could look at, and this is the baseline of chromosome breaks in humans. And he found one guy with huge levels of chromosome breaks. He followed him for a year, and then he had stumbled on this folic acid deficiency breaks chromosomes. So he measured this guy's folic acid. He had practically no folic acid. And so we gave him a pulse of folinic acid, which is a reduced form of folic. The chromosome breaks came down, and then they started going up again. He put him on folic acid permanently. And McGregor and I went to Kaiser, and we found lots more people. There are lots of people out there breaking their chromosomes because they weren't getting folic acid. And when I looked up the level, it was half the pool. So I said, wow, this stuff is really interesting. This part per billion of pesticide, we've been publishing lots of papers that has nothing to do with cancer. It's all hype. And you don't want to dump pesticides everywhere or have chemical companies put garbage out the back door, but it's not a significant source of cancer. Anyway, so we had a paper, and Fenwick in Australia had a paper comparing radiation with folic acid deficiency. And people are worrying about incredibly tiny levels of radiation. We're going to spend $6 trillion cleaning up tiny amounts of radiation around Hanford. We're talking about amounts that are barely different from background. They could have given a vitamin pill to everybody in the Pacific Northwest, and they would have been $6 trillion ahead, and would have been way ahead in public health. Anyway, iron. So we heard about iron, and Fernando Dietary, who's a professor of nutrition at Berkeley, knocked on my door one day and said, the World Health Organization, I can't stand it, they're giving people too much iron. And so we went through, he got me all excited, and his graduate student, Mitch Knudsen, and one of my postdocs, Patrick Walter, did the whole range of iron in mice. And what we're measuring are all sorts of things that go wrong with mitochondria, with too little iron or too much iron. The mitochondria pour out oxygen radicals, you get massive DNA damage, they're inefficient, and you get it with either too little iron or too much iron. But this is 2 billion women and children in the world, and this is a few men who are eating too many steaks. So, and this is where you should be. So you don't want to overdo these things. Mae West said too much of a good thing is wonderful, but she was thinking about sex, not micronutrients. So if somebody says iron is good, you don't want to pig out on too much iron. Anyway. And so Joyce McCann, who's a very smart woman, she used to be a postdoc with me about 40 years ago, and she came back to the Bay Area, and I knew she was super smart, so I hired her. And she's written 5 big reviews on brain development. During the first 2 years of your life, and when you're a fetus in the first 2 years of your life, you're making 1 trillion neurons, and each neuron has 1,000 connections. It's one of the most complicated things in the universe, the human brain. You don't get enough iron. The rat pups don't do well on IQ tests. The mouse pups don't do well on IQ tests. And the kids don't do well in school. But human studies are always very iffy and difficult. But she did a beautiful review on all of this. And her recent review, the 5 of them that she's written, her recent reviews on vitamin D. And we'll talk about that. Now, Emily Ho, a postdoc in lab, was looking at human cells and culture. These are primary human cells. And she looked at zinc deficiency, and they pour out oxygen radicals when they're zinc deficient. They're pouring out all these oxidants, and you get lots of DNA damages measured by the Comet assay. And she also showed you knocked out P53. And then, so anyway, we were showing zinc deficiency and biotin deficiency and iron deficiency all give you DNA damage and oxygen radicals. And Hani Atamna, a wonderful postdoc in my lab, worked out why. So why did so we were getting oxygen radicals, we thought it probably was coming from the mitochondria. It turned out you were interfering with heme synthesis. And when you do, the first thing that goes is heme A, which is a decorated form of heme. It's only used in one place in the cell, in complex 4 in the mitochondria, and that's the first thing that goes out. So there's a big excess of complex 4. And complex 4 is adding the 4 electrons to oxygen. When you're short of heme, then you get a decrease in the amount of complex 4, and you start pouring out oxygen. The reason there's an excess of complex 4 is to keep oxygen levels down. And so now we've shown that all kinds of things, iron deficiency does that, pyridoxal phosphate deficiency does that, this enzyme has 8 zincs in it, zinc deficiency does that, and we're slowly filling in this table. Here's complex 4, and the excess to keep oxygen radicals down. And here's human cells in culture, you make them a little biotin deficient, they fill up with oxidants. Biotin is a mitochondrial coenzyme that replaces the intermediates that come off the Krebs cycle. So, and since heme synthesis is a major pathway, and one of the main ways you're taking intermediates from the Krebs cycle, you need the biotin to keep replenishing. And it also makes human cells in culture senescent. So when you're biotin deficient, they senescent. When you're magnesium deficient, they senescent. When you're perioxide deficient, based on SRE. Those are the only three we've done so far. This shows this table. We published a paper, Hani Atamna published this paper. I did all this work on biotin. The papers come out. And, another postdoc, Chinu Askari, is doing it on perioxin. He's filling in all these blanks. And we are going to fill in the rest of the blanks. So at least for these micronutrient deficiencies, we understand the mechanism. And this show, and David Kilday in the lab is working on magnesium, since so many Americans are short of magnesium. And magnesium deficiency ages cells faster. And he's shown you get lots of DNA protein cross-links when you're magnesium deficient. We've shown human platelets are full of these cross-links. So we think that's going to be a good way of measuring micronutrient deficiencies in people. We haven't shown it for more than magnesium, but I suspect all the micronutrient deficiencies in people. And so I looked in the literature once I had this idea about triage, this idea that it's long-term versus short-term, and there's all this stuff in the literature. Calcium deficiency breaks chromosomes associated with cancer, folate deficiency associated with 4 or 5 types of cancer and breaks chromosomes, vitamin D deficiency. There are 100 new papers about its big risk factor for cancer. Magnesium deficiency breaks chromosomes and has been associated with cancer. Zinc deficiency, esophageal cancer in rats and humans. Potassium deficient, vitamin B12, selenium, omega-3s, niacin, choline. And this is just what I got off Google in a half-hour worth of work. And now we're digging into all this and writing reviews. Every time we turn around, we find more evidence saying that's how nature works. There are binding constant differences to explain why some metabolism goes out over others, and it's been built into our system over all of evolution. So the next step is to do human clinical trials, and we made a bar having all the micronutrients and fiber and all the things that these people are lacking, and it takes away your appetite. Because I think some of the hunger of obese people is that if they're missing magnesium, the body makes them hungry, so they eat a little more so they get some of that missing magnesium. Nature would rather have you fat and fertile than thin and non-fertile. Anyway, this was back in end of 2006. I had the first iteration of this idea, and we're writing up another one now because we have a ton more evidence. That's not a good one. So the immune system suddenly hit us. There's a paper that lists 6 things that go wrong with the immune system with age, mostly adaptive immunity. And when you start digging in the literature, people have tested a few things, and there's evidence for this, evidence for that, evidence for this, and this on micronutrient deficiency. So, and that's long term, because if your adaptive immunity is out, you die of a more severe infection 5 years down the road, just the way you die of cancer 5 years down the road. Nature doesn't care when you're starving for something. And mitochondria pouring out more oxygen, nature doesn't care because it's long term. It wants you to survive so you can reproduce a little, and maybe you'll eat something that gives you the missing micronutrient in between if you don't die first. Anyway, we actually, Chantal Courtemanche, who is a French-Canadian postdoc in my lab, terrific gal, had shown that the immune system goes out when you're folate deficient, and what she found is the CD8 cells go out relative to the CD4s. And I said, what's that mean? I don't know what that means. Well, you might as well publish it. It's an interesting observation. So she talked me into it. When we published it, I didn't understand it at all. And then when we were doing this new stuff, it all became crystal clear. The CD8s going down relative to CD4s, one of the things that happen when you're short of micronutrients. It's long-term stuff rather than short-term. Anyway, so what about people? This says you're 57 years old, I'd like to get that down a bit. And you have to do studies in humans, not only in rats, and that's what we're in. Humans are, of course, very difficult. But anyway, we're doing clinical trials now, and we hope to show that micronutrients lower your DNA damage, improve your adaptive immunity, and make your mitochondria so they don't pour out so many oxygen. And I'm going to be 80 in December, and maybe I have 5 good years left, I don't know. But anyway, that's what I'm trying to do before I pack up. Anyway, thank you.